Okay, everyone, if you'd please have a seat. My name is Brent Brockmeyer. I'm with Vaughn Pools in Jefferson City, Missouri. We'll go through the housekeeping here, as I'm sure you guys are getting tired of this, but this is one of the things they're told that we need to do. I want to welcome you to the conference. This particular session is the effect of cyanuric acid on inactivation of Cryptosporidium. Need to ask you to turn your cell phones off, or to at least put them on vibrate and sit towards the back of the room so that you can exit if you must. The emergency exits out the door and either way at a 90-degree. The restrooms are out the door and to the right, and before you step outside, again to the left, unless you want to use the third cactus on the right. And of course, there's no audio recording or video devices. The presenter this afternoon is Dr. Vincent Hill. He is a research environmental engineer for the Waterborne Disease Prevention Branch at the Center for Disease Control in Atlanta, Georgia, National Center of Emerging and Zoonotic Infectious Diseases. He's the author of over 50 peer-reviewed journal articles and scientific reports, and he's also a team leader for the Water Sanitation and Hygiene Laboratory team. I give you Dr. Hill.
Thank you. All right, good. You can take that because everybody can hear me, right? Thumbs up in the back. Great. Okay. All right. Well, as Yogi Berra might say, if he was still alive, this is deja vu all over again. I feel like I've done this before, and I did this morning. So it may be deja vu for some of you as well, since some of my slides are pretty similar where they overlap with the previous study on chlorine dioxide. But bear with me if that's the case and you're seeing them again. Okay, so it's a pleasure to be with you. I'm going to basically talk about research studies that we've done at CDC on hyperchlorination or chlorination under conditions where cyanuric acid is present. So this is one of the slides I showed earlier, just kind of going over some of the typical pool disinfectants. Chlorine-based, bromine-based are primary disinfectants. That means the disinfectant is present in the pool water itself and immediately is active against pathogens that are excreted or get into the water. So it's immediate, you get immediate treatment. Versus ozone, and in this slide I actually added, I kept UV in here. I didn't have it this morning. It's not really a chemical disinfectant, but it is a secondary treatment technique in which the water has to be pulled out of the pool essentially and passed through a treatment technology, a treatment unit, in order to get that disinfection. So that's very different than a primary disinfectant. So focusing in on the chlorine-based disinfectants, I'm going to focus in this talk, of course, on stabilized chlorine, which is one of the families. It's basically the stabilizers added in addition to free chlorine in the pool. So cyanuric acid, or as you might hear me say, CYA, in this talk, and this is the other CYA, not the CYA you may be more aware of, or when you're covering various things, but CYA is an organic molecule, and it has this ring structure, and that ring structure can have chlorine molecules attached to it, and it stabilizes the chlorine and prevents UV degradation of the chlorine. I'll talk a little bit more about that in a few slides. It's been used in outdoor pools for a long time, since the 50s. It itself, cyanuric acid, has no disinfection properties. It's the chlorine that has a disinfection property. So, it's often present, and this is, there's a great range, but it can accumulate in pools to fairly high levels, upwards of 100 to 150 milligrams per liter. So there are three forms available for pool use. There's cyanuric acid, or CYA, which you might consider a conditioner because it's added in separately to a chlorinated pool. And then you've got dichloroisocyanuric acid, or dichlor, I'll definitely say dichlor, or trichlor, which are added, they're already combined with the chlorine, and so it's added as one molecule into the pool. So here's some CYA chemistry. So up to 3 chlorine atoms can combine with a CYA molecule. So it forms a temporary weak bond, and the chlorine, as I said before, is protected from degradation. So on the left side there, you've got the 2 chlorine atoms with the cyanuric acid molecule, so that's dichlor, so 2 chlors, di. And then the 3 chlorine atoms on the right, that's trichlor. Essentially, that molecule added to water creates an equilibrium, and hypochlorous acid is produced. So that's the hypochlorous acid, and that is available for disinfection, whereas the CYA molecule really has minimal disinfection power on its own. So the chlorinated isocyanuric kind of behaves as sort of a reservoir for free chlorine. And the chlorine is then released into the pool as hypochlorous acid is depleted. It's released from the molecule, that is, as the hypochlorous acid is depleted in the pool. So free chlorine is eventually completely depleted, and so you have to add more chlorine to the pool as the chlorine dissipates. But the CYA remains and will build up over time. Especially if dichlor or trichlor are added to increase the free chlorine concentration, then you're also adding more of this cyanuric acid molecule. So the method that we have used at CDC for measuring cyanuric acid and free chlorine, so we use the Dpd method. So that's a standard method for measuring chlorine. And basically, the amount of chlorine, the chlorine reacts with this Dpd molecule, creates a pink color, and the more chlorine that's present, you get more of the pink color, and then that's measured by a meter and reads out as the free chlorine concentration. So the chlorine that's associated with the chlorinated isocyanurates is identified as free chlorine in this test. So even though it's bound in that molecule, you still get it reading out as free chlorine on your test. So that's important to recognize. CYA itself, if we're measuring that separately, we measure it by this melamine turbidimetric method, which is readily available through a company like Hach. So I just want to step back and kind of put some of the work that we're doing kind of in context with what's been done by other researchers for quite a while. So since the 1960s, researchers have looked at cyanuric acid and its effects on chlorine inactivation. So early on, as most of this research was focused early on bacteria. So bacteria like Streptococcus faecalis, which is now called Enterococcus faecalis, Staphylococcus aureus, or staph, were investigated by Anderson as early as 1965. And basically that research group found that the more cyanuric acid that was present in their experiments, whether it was 20 or 50 or 100, they needed more chlorine than in the reactions in order to achieve the same level of inactivation of bacteria. So with increased concentration of cyanuric acid, they had to add more chlorine in order to compensate or overcome that effect. Then a little later, still in the 60s, looking at E. coli and other bacteria, again, this research group found that 50 milligrams per liter, 50 ppm, of CYA retarded disinfecting action of calcium hypochlorite by a factor of 4 or more. So they're starting to building in as early as the 60s some indication that cyanuric acid slows down the inactivation kinetics of free chlorine. Again in 67, this one with Streptococcus faecalis, again found that at levels of 25 to 100 milligrams per liter, CYA increased the time required for 2-log inactivation by free chlorine. And then the last one from the 60s, this one is Pseudomonas aeruginosa, so it's a slightly different bacterium, found that at the lower free chlorine concentrations, CYA caused longer contact times required to inactivate this particular bacteria. So then jumping in, so that's all bacterial data, right? And so jumping then further into the 1980s now with viruses. So Yamashita et al. Looked at enteroviruses and adenoviruses and found that to achieve a 3-log or a 99.9% inactivation of those microbes, those viruses, by free chlorine, that it was prolonged by 5 to upwards of 30 times by the addition of 30 milligrams per liter or ppm of cyanuric acid. And they reported that the rates of the inactivation were affected to the same extent by cyanuric acid in swimming pool and tap water. So they're looking at different kinds of water. So they found the same effect. So now we've got some data on bacteria, now we've got some data on viruses. And then to add to that, so we kind of knew about that when we first started doing this work at CDC, so we decided, well, let's look at bacteria ourselves and viruses ourselves. So we did a little bit of work with staff and looking at a range between 0 and 100 milligrams per liter, and found that when we had 0.5 milligrams per liter free chlorine, the FC there means free chlorine, at those water conditions, that the higher cyanic acid that we added in, the CT values, or the concentration and time needed to achieve the 3-log inactivation, increased with the higher concentrations of cyanic acid. And then the inactivation times were proportionally increased up to from 1 minute down up to 13 minutes with higher cyanic acid levels. And then when we looked at MS2 virus, which is this, it's an enteric virus, it sort of uses an indicator for pathogenic viruses. The data set's a little smaller. It's only, we're going to add to this, I think. It only goes up to 25 milligrams per liter, but we saw a similar effect with the increase in CT and the inactivation times associated with the higher levels of cyanic acid. So, based on this research, the cyanic acid has been shown to reduce the efficacy of chlorine against bacteria and viruses. But in a worst case scenario, the contact times required for inactivation were only extended by minutes. So bacteria and viruses die off so readily that in some ways it didn't really matter so much because chlorine is so effective against them. So for many years it wasn't really a big question, so there was a trade-off. So the benefits of the stabilization that you got with cyanic acid was considered useful in pools, for especially outdoor pools where there's a lot of UV exposure. So, but now the question is, with the advent and the issues related to parasites and Cryptosporidium, is what about parasites? Do we see a similar thing with them? And we know that they're more chlorine tolerant than the bacteria or viruses. So where a slowing down of inactivation on the scale of minutes, it might be okay with viruses and bacteria. With parasites, because they're so tolerant, then chances are that it might not be the same situation. So one of our parasites is Giardia, and it has a relatively higher 3-log inactivation or tolerance to chlorine, about tenfold higher, so to speak, versus bacteria and viruses. But really, the big concern is Cryptosporidium. And it's well established now the extreme chlorine tolerance that crypto has. Depending on the isolate that we've used in different studies, we get a range of these CT values from between 10,400 to 15,000. So orders of magnitude higher than the bacteria and the viruses. So obviously then we're going to focus in on crypto. And just to step back a little bit again, if you've heard this earlier, bear with me, but one of the basic physiological aspects or characteristics of Cryptosporidium is it has a really thick wall that protects the 4 viable sporozoites, or the infectious life stage of Cryptosporidium, that then when they are released, they cause the infection in the human gut. So you have these sporozoites inside of crypto, but that thick wall really protects those sporozoites from being damaged by disinfectants, chemical disinfectants. And we know that when a person is sick, they can shed up to 1 million Cryptosporidium oocysts per gram of feces. So you can get a lot of crypto oocysts shed in a diarrheal incident. And only as few as maybe 10 oocysts need to be ingested in order for a person to be sick. And we know that there's incidental ingestion during swimming, whether adults, kids will swallow a lot of water. And so at those levels, with those kind of shedding and the high infectivity of the oocysts, it's no surprise that crypto is a big issue, considering its chlorine tolerance. And as I said, if you ever go to a CDC talk, you always see surveillance data and from our national outbreak reporting system, and so this is no different. And so basically it just puts it in context that we know that recreational water outbreaks have been increased in reporting over the years, and along with that, in the blue, are those associated in treated recreational venues associated with Cryptosporidium. And the disease called Cryptosporidiasis. So we know it's a big issue and it's a major proportion of our overall outbreaks. And then of those those pathogens that cause gastrointestinal illness in these treated water venues. You know, sure, there are some chlorine-sensitive microbes that will cause outbreaks, but those are mostly due to poor pool operation and maintenance. Really, when crypto shows up and somebody's sick and they swim, there's nothing about pool operation, you know, that can deal with it because it's so chlorine tolerant. It's just going to be readily an issue that it's going to be infectious and a risk. So that's why there's a huge proportion of our outbreaks caused by crypto. So why study cyanuric acid in crypto? It should be pretty obvious by now. So cyanuric acid, of course, is widely used in the industry. And we know that when there are diarrheal incidents, or we know that crypto contamination is likely to have occurred in a pool, we have CDC's recommendations for emergency response to those situations that currently require a 12.75 hour contact time for 3-log inactivation at 20 milligrams per liter of free chlorine. So just using chlorine alone. But the previous work suggests, right, with bacteria and viruses, that there's decreased efficacy of chlorine against crypto when cyanuric acid is present. So that raises the question, what's the extent of the effect? And that's what we wanted to answer. That's the big question, is what do you do if you've got cyanuric acid in your pool and you're trusting this remediation protocol to return your pool to a safe status for people to swim in again? Can you really trust that the chlorination, hyperchlorination protocol is really going to achieve the performance that you are expecting? So early on at CDC, we did some work in this area. Joan Shields led that effort back in 2008, 2009, and published this paper. And in her work, I'm going to go over her work a little bit. She also looked at the differences between Iowa strain and Maine strain. And so this is going to get a little technical, but it's pretty important to just kind of understand real quick. The Iowa strain, which was used in that initial CDC cyanuric acid study, has been used for a long time. It originated in the early 1980s from Ames, Iowa. That's why it's called the Iowa strain. Sometimes you'll see it's called the Harley Moon strain or isolate. And it was passaged in calves as well as mice, and it's widely available. There are commercial sources of it, and it's easily obtainable. So that's why a lot of the research that's in the literature uses this Iowa strain. At CDC, we have and we propagate another strain that was isolated in Maine from a human outbreak. So not calf, it wasn't isolated from a calf, it was isolated from humans. So it's different in its origin that way. The source was an apple cider outbreak, associated outbreak. Now, we do passage it in calves now. There is no other way to reproduce Cryptosporidium oocysts, so we have a calf facility at CDC, and we make our own oocysts that way. And we've found then in research that it's more chlorine tolerant than the Iowa strain. So it's the same species. It's still Cryptosporidium parvum, but there have been some documented differences between them. So I'll try to kind of keep that in mind when I'm talking about the work. So Shields' work was all done with the Iowa strain early on. And so in the first body of data that was reported in that paper, the first experiment was done under kind of standardized pool conditions that we established, again using this Iowa isolate. And after 10 hours, with a free chlorine concentration of about 20.7 milligrams per liter, we could achieve about a 3.7 log inactivation. But when cyanuric acid was present, with the same basic free chlorine concentration, we couldn't get even a 1 log inactivation, only 80% inactivation there. So based on the free chlorine controls, we felt like which calculated out the concentration of the 20.7 times the time allowed to get to the 3 log inactivation, estimated a CT value of about 10,400, which was in general agreement with the literature and expectation with the Iowa isolate. So we felt like the data was solid, except that we found that when you had cyanuric acid in the reaction flask, it really slowed down the inactivation of crypto with chlorine. In the second set of experiments that were reported in that paper, she did some things like decrease the pH to 6.5 instead of up to 7.5, because we all know maybe that free chlorine is more active at the lower pHs. And then also looked at using higher free chlorine concentrations. So instead of 20-ish milligrams per liter, looked at 40. So the first row there is the same basic experiment that was reported earlier, 50 milligrams per liter cyanuric acid with the kind of typical hyperchlorination level of 21 milligrams per liter of free chlorine. But then you see a log inactivation only 0.6 or 75% inactivation after 10 hours, and not only just barely getting to 1 log after 24 hours of exposure. Sure, when you double the amount of free chlorine in the reaction flasks, you can get higher levels of inactivation. Not too much after 10 hours, but maybe up almost 3 log. So almost achieving the 3 log goal that we have for hyperchlorination for emergency response, but it takes 24 hours to get there. So that's not a really good protocol to recommend to people. And you can see the associated CT values calculated down there were 86,000, 67,000, et cetera, for different conditions. And so that is well above the 15,300 that's currently recommended. So none of this, it's all kind of provided an evidence base, but really no guidance forward on how to solve this problem or really kind of characterize it better. So, however, this was useful, though, because it was the first study that kind of shined a light on this issue for cyanuric acid's effects on chlorination for parasites, for Cryptosporidium. So that was really useful, but we saw that this data really fits within the literature that had been established for bacteria and viruses, that cyanuric acid is associated with slower inactivation times of pathogens, microbes with free chlorine. Okay. So that led into the need and the prioritization of the CDC to do some additional work on cyanuric acid because we wanted to characterize things better. We wanted to better understand the effect of cyanuric acid on free chlorine and activation of Cryptosporidium, and we wanted to help provide more solutions. We didn't want to just say, hey, there's this big problem here, somebody figure it out. We wanted to help kind of push the science along as much as we could. So the focus of the studies I'm going to kind of really dive into here with you is to basically generate 3 log CT values for crypto inactivation at multiple cyanuric acid levels under remediation conditions. So really primarily looking at 20 milligrams per liter free chlorine, but also potentially higher levels as well. And then so the approach was then to measure the free chlorine concentration over time, measure the crypto concentration over time until we got to that 3 log level. We didn't want to fall short. We wanted to get to that 3 log level because that's what all the recommendations are based on for response. And we wanted to use this crypto main isolate. So previous literature had said that it's the tougher microbe to kill with chlorine, so we wanted to be, wanted to see what the worst case scenario was, if we could. So if you saw my talk earlier, it's the same basically the same setup it's a jar test, the jar test approach. In this case, of course, things are different. We got different chemicals involved, but we still have some controls. So we still have an oxidant demand-free water control, which we always include to basically, when we'll add Cryptosporidium to that, and we track that over time just to make sure that if there's die-off of crypto, that is it due naturally to the crypto dying off. So there's no disinfectant added to those flasks. It's just tracked to see. We shouldn't see any Cryptosporidium reduction in those control flasks. Then we include a 20 milligram per liter or kind of typical hyperchlorination flask. And that's done basically to make sure we have some kind of way to relate our data from this set of experiments to previous experiments that have been reported in the literature. And similarly, and we wanted to also look at 40 milligrams per liter. That had been reported earlier and wanted to kind of bookend what hyperchlorination approaches might include, although 40 is a lot of free chlorine to use in a pool. And then, so those are our 2 free chlorine conditions, but then the other 2 flasks then had varying concentrations of cyanuric acid. So we would repeat this over and over with different levels of cyanuric acid. And those were basically upwards of 100 milligrams per liter at the high end, down to 8 milligrams per liter on the low end. And then 16 and 50 in between. So we had 4 levels of cyanuric acid that we were investigating. So as we usually do, then we cover the flasks. They're always continuously stirred, and we maintain them at 25 degrees C throughout the experiment. So the pH, initial target pH, was 7.5, and then we did measurements using standard methods for free chlorine, cyanuric acid, and ORP as well to give another measure of oxidation level in the different reaction flasks. So we would take samples over time, so not just taking samples at time 0 and at the end of the experiment, but taking them all the way through the experiment at different time points so that we get nice curves for an activation study. So we collect these samples, and then we'd go and perform the infectivity assay. And when I say infectivity assay, what that basically means is we've got basically cells, mammalian cells that kind of reflect the gut cells that we have in mammals. So, but they're not human cells. They're actually they're dog. MDCK means dog cells. They're kidney cells that we put. But they make a little monolayer in these flasks, and they're receptive to the Cryptosporidium infection. So we get these samples, we neutralize the chlorine using sodium ethyl sulfates, concentrate the cysts down to a very small amount, induce exsistation, and that's a kind of a technical term, but basically what it means is inducing those oocysts, those tough oocysts, to open up and release those sporozoites into the flasks that have the cells that could be infected. So they get excisted, the sporozoites are released. If the Cryptosporidium is still viable. If it's dead, that doesn't happen. And so then we do the infectivity assay on the cells. We incubate them for a while to let the infection progress, and then analyze it further. So we process these slides. So again, they have these cell monolayers on the slides. There's sort of a pink color over there. We wash off all the stuff that we don't want to stay on the cells during the infectivity and labeling process. And so we use this fluorescent antibody. So just like we have antibodies in our bodies here that react to pathogens that we're exposed to, right, to remove the pathogens and limit infection, we have those antibodies in test tubes, and we can take those antibodies and we can attach them to fluorescent chemicals, and those antibodies that we use are specific to Cryptosporidium life stages. So if there were viable Cryptosporidium in a sample that were on those monolayers, then we'll see it light up, the different points at which there were infections, and we'll be able to see that. And we see that using microscopy. So we use these fluorescent microscopes to capture images of these fluorescent points in which there were cell infections, if there were viable Cryptosporidium after the experiments. The different life stages that are created when the cells are infected, there are different life stages. All of those are going to fluoresce with these antibodies. So we have, we've taught the microscope takes the images, and we've got software that we've taught, so to speak, to recognize the size, the shape, and the type of fluorescence that is associated with those different life stages. And so the computer captures all that information, and we can use that data quantitatively to calculate the level of inactivation that occurred during the experiment. The image here on the left side is what the microscope sort of sees. It's kind of the starry night. Each one of those bright spots there quite possibly is a point of where a life stage is, where there was infection. And so the microscope will capture that, and then the software will say, okay, some of that is just junk, but I think, being the software, that those are all life stages, and then it'll count those up. And then we have physically gone through, Jen Murphy, who is the co-author of this, physically went through and visually double-checked things and made sure that the software wasn't just kind of getting confused about certain things. And so for quality control, went through every single one of these images. For this particular study, I can't remember how many there were, but I mentioned in the chlorine dioxide study that there were like 17,000 images that she had to go through. I think it was actually more for this study. So it's very labor intensive, but that's what we do to calculate the level of infection associated with the different samples that are collected through the experiments. So, and here's the output from some of the output from the data from the experiments. So these are basically data to calculate 3 log, again, 99.9% inactivation values, CT inactivation values for crypto at these kind of ideal water conditions. On the left column there is the average free chlorine concentration. So the top rows there reflect kind of what I say typical hyperchlorination conditions of around 20 milligrams per liter. The top row is there's no cyanuric acid in those flasks in that data. And it took basically about 8 hours average time to achieve a 3-log inactivation at that concentration, which calculated out to about 10,500. So that's really well in tune with the range of, because there's a range there, it's 9,200 to 12,000. So it's a little bit lower actually than was reported in the SHIELD study, and that's something that maybe we can talk about later, is maybe as we collect more data, that 15,300 might be warranted to revise that downward if we collect more data like this from Maine, because this is at 10,500. So it might be that something less than 10 hours might be just as appropriate for achieving a 3-log inactivation of crypto. So that's a little side comment. But if you look then with the average CYA concentrations, the next row down is 8 milligrams per liter. Then you go from 8 hours to 14 hours required to get the 3-log inactivation. When 16 milligrams per liter CYA was present, now we're up to 27 hours with the associated CT value up around 31,000. So pretty clear effect there of CYA on hyperchlorination with 20 milligrams per liter. Then we decided to look at the 40 milligrams per liter as well. Again, the top row there is without cyanuric acid. The CT value there calculated was 12,400, so still in that same range that we were kind of expecting without having cyanuric acid present. For those experiments, instead of 8, they averaged out to be 9 milligrams per liter cyanuric acid. But again, there's an increase in the time required, not as much actually as the earlier one, because now we've got 40 milligrams per liter of free chlorine present, right? So the overall times to achieve the 3-log inactivation, of course, are going to be shorter. So even upwards of where there's 15 milligrams per liter cyanuric acid present, we can still achieve the 3-log inactivation within 8 hours or so. So but still, that's a lot of free chlorine to use for hyperchlorination, I understand. So, but at least there's some ray of hope, so to speak, that there are other maybe approaches, or there's coming maybe a mix of approaches in which you can maybe hit a certain range. But still, even with that data at 40 milligrams per liter, there's still a transition there with higher times required for inactivation. And those cyanuric acid levels are pretty low, right? I mean, 15 milligrams per liter is pretty low. It's not typical. So next set of data here is with 50 milligrams per liter. So things get worse. And in fact, the CTs, we couldn't calculate CTs for 3-log inactivation because we couldn't achieve a 3-log inactivation within a reasonable amount of time for the experiments. And we're talking days that we'd have to go out. And we just didn't get there with these experiments. So these are 1-log CTs. And the top level, again, is your typical hyperchlorination levels of about 21 milligrams per liter. And you just go from 0 to 48, and you see it goes up from, for a 1-log. Now remember, there's a 1-log, not 3-log. So from 2.7 hours to 62 hours, just to achieve 90% inactivation when there's 50 milligrams per liter. And even 50 milligrams per liter is not outrageous for a pool. Now, if you look at the 40, though, you can see there, okay, well, if that 46 is present, of cyanuric acid, it's only 17 hours, so it's still a long time. But it seems more reasonable, but still, that's a long time for doing one of these remediation protocols. That's a long time for a pool to be down. So I don't even know at the higher levels if even trying to use the really high levels of free chlorine for hyperchlorination is a feasible approach. So again, we could not meet the target 3-log inactivation level for crypto within, say, a 24-hour time frame when we looked at about 50 milligrams per liter cyanuric acid. And we did look as well at 100 milligrams per liter. And so for this, we couldn't even calculate CT values. The data, we just couldn't get to 1 log repeatedly to even do a 1 log CT value. So these are basically just kind of, they're just bullets here. So we did achieve a 0.8 log reduction, or an 84% reduction when there's 20 milligrams per liter. So when you're doing hyperchlorination, 100 milligrams per liter in there, after 144 hours, so 6 days of exposure, we still couldn't get to 1 log. So this is, just sort of academic in some ways. It's just showing you how extreme the problem gets when there's a lot of cyanuric acid present in the water samples. when we used 40 milligrams per liter free chlorine, we were able to get to the 0.8 log reduction, or 84% reduction, in 24 hours. So it went from 6 days to 1 day. And then we got to a 1.4 log reduction in three days of exposure. So again, it's sort of academic. Nobody's going to want to do that. And it's still not even 3 logs. And so again, we couldn't calculate a 1 log inactivation or CT values for free chlorine when 100 milligrams per liter is present. So here, these are in the paper. So this is all published now. Came out earlier this year, and there's the source down there. So these are basically figures from that paper. The top row there is, so again, these are log inactivation or percent kills versus time when we're using 20 milligrams per liter free chlorine from 0 to 50 milligrams per liter cyanuric acid. And the 0 is the sharper curve. So that shows how fast we were getting inactivation, the one all the way on the left, when there's no cyanuric acid present. Then the next curve is with 8, the next curve is with 16, and the one way at the top there is with 48. So thank you. So you can see the progression, so to speak, into slower inactivation times. And these differences were significant in the data set after we get to 2 hours. So from 2 hours onward, everything is significantly different. Now when we go up to 40 milligrams per liter, sort of interesting in that the 40 milligrams per liter bunches things up a little bit more. Still, though, the leftmost curve or line is without cyanuric acid, so that's the fastest. The next one is with 9 milligrams per liter CYA. The next one's 15, and the top one there is with 46. So, and again, although they're bunched up there, statistically, even after about 2 hours of going through the data set, they start being, the data between those different cyanuric acid levels becomes significant. So there is a significant effect of cyanuric acid on inactivation rates. For the 100 milligram per liter CYA level, we couldn't even do all that fancy statistics. This is really just observed data with lines kind of drawn through the data. So the fastest, again, the fastest curve there, and you can see at the top, we're in hundreds of hours to get even out there to where 2 log might be, and we didn't even get there for some of the experiments. But the one on the left, again, is CYA with 0 CYA. And the next curve is with free chlorine at 20, and then I'm sorry, I'm getting myself mixed up. The one on the very right-hand side is actually natural die-off of the crypto oocyst. So if you take things out so far, 300 hours, crypto just starts dying off. It's sitting in a, it's sitting in an environment it doesn't want to be in, and so you get natural die-off. So a lot of the inactivation at this level was really due to, we had to take the experiments so far out, was due to natural die-off, and that also kind of made it difficult to really evaluate the data. But the 2 left ones are the differences between 20 milligrams per liter free chlorine, which is the center one, and then 40. So with 40, again, you get a faster kill, but on a scale of hundreds of hours, really. Okay. So I know time's running out, but for those who like to see the ORP data, wanted to throw this in there because you can see that when no cyanuric acid is present and there's 20 milligrams per liter, we're at that 770 millivolt level. Even with 8 milligrams per liter cyanuric acid, you don't see much difference. But then you start seeing progressively a suppression or a decrease of the ORP with higher levels of cyanuric acid. You see the same basic effect when 40 milligrams per liter free chlorine is present. So, some conclusions. So the CDC, our research on cyanuric acid, like I said before, is consistent with historical literature. The previous literature was all on bacteria and viruses. This is on parasites, but really the general conclusion that cyanuric acid decreases chlorine disinfection rates pretty much fits within the previous literature. Hyperchlorination might achieve a 3 log inactivation of crypto at the low cyanuric acid level, so less than 20 milligrams per liter. Within 24 hours, but the higher free chlorine levels are going to be needed to really, if you're at that level of cyanuric acid, you're going to need more free chlorine probably to achieve a 3 log inactivation. We couldn't obtain a 3 log inactivation of crypto when cyanuric acid was present at 50 milligrams per liter or higher. And so I mentioned the other sort of a side comment about potentially decreasing the CDC response recommendation based on the main isolate hyperchlorination without cyanuric acid data, but I won't focus on that so much. I do want to say though, last slide here is like, so we understand that there's that this still kind of suggests that there's an issue with pools that have cyanuric acid. That how do they really achieve the expected Cryptosporidium kill when there's a fecal incident? So that issue still remains, and I don't know that we're really going to achieve it with hyperchlorination on its own. So there are other potential approaches. One of the ones I talked about this morning was potentially using chlorine dioxide. We haven't done that work yet with water containing cyanuric acid, but that is a project that we received some funding from NSPF to look at, and so hopefully next year, by this time, we'll have results that I can present on that. And we heard earlier, I put a question mark here of whether chlorine dioxide could be used for normal operations, but I've been hearing that even in Australia that it is potentially being used, I think, for normal operations. So even that might be possible. It might be just a bureaucratic blockage for why it's not being adopted in the United States. In addition, we're looking at other water treatment technologies, looking at sand filter aids and clarifiers as collaborators with James Ambergi at UNC Charlotte. So the crypto removal that way. And then we're interested in looking at other technologies like UV or ozone. We just haven't been involved in those studies yet. So thank you all for being here. Appreciate it.
Thank you, Dr. Hill. That's the first presentation on crypto and Cyanuric that I could actually follow.
Oh, good. Thank you.
That was fun. Questions?
Doctor, your study showed a pH of 7.5 or 7.6 in that whole thing. Did you do any at lower pHs?
So we haven't. And sitting in some other talks, I've been wondering if we should. The problem is like every time you take a parameter like that and turn it into a variable, it's just doubling or tripling the amount of work. And basically both of these studies took about 4 months of lab work and about a year overall to manage all the data and analyze it and everything. So I'm a little loath to say we would do 7.5 and 7.2 and 6.8, as a true variable. But I wonder if 7.5 is a really good pH, should we lower it to 7.2? Is that more typical? I mean, maybe it's in the higher end. I don't know, but I think we probably won't do a whole lot on varying pH. Really? Okay. All right. Thank you.
So Vince, really good talk. Over here.
Hello.
So I'm wondering if the bound chlorine has any effect at all on crypto, and I'm wondering actually if your data would allow you to evaluate that. Specifically, I think you could probably calculate how much is bound and how much is not bound, and if you assume that the bound material is ineffective completely, then using the unbound free chlorine, you could compare that with your data from just the free chlorine experiment and see how well they match up. Have you done that?
We haven't, and we've got a head nod up here because it's been suggested to us before. Yeah, so I think it's a really good, it's a good comment, and I think it's something we should do. We haven't done it yet is the short answer. Yeah.
Yeah, I did that with all the data. I did that with the Shields data too.
Oh,
this data
there we
is
go.
consistent with that too. It tracks for the HOCl and within error, because there's measurement error of the cyanuric acid and such, the chlorine bound to cyanuric acid is effectively to be ignored. It has very, very minimal.
So Richard's you're gonna publish that?
No,
You're gonna publish that when?
I mean, I'll send it out to anybody. That's not peer reviewed research, right?
Thank you. But we can follow up on that some more too.
Are you aware of any crypto data with bromine?
I'm not, and I've been asked, so Michelle Lavza has asked me a number of times that we should try to get a project with bromine. We haven't developed a proposal for it. If it's something that's of interest, whether NSPF, there's not a whole lot of funding sources, and it's not something that we have the time to put somebody onto. So it's like often when a project presents itself, we'll do it. But so maybe we can talk more about that. Not that you you're going to be funding it. But just to know more about the kind of the chemistry of it and the kind of the need for it. So, yeah, but it's been suggested before. Thank you. With bromine, yeah, it would be faster, but you have the byproducts to manage.
Oh, okay. Hi, thank
Hi.
you for the talk. Have you done any testing with different water temperatures?
Again, no, we haven't. It's sort of the same issue with the pH in that it's sort of just what we can do with the work, the staffing time that we have and everything. Yeah, so we chose 25 as just sort of, generally speaking, kind of average temperature. It's like 77 degrees Fahrenheit for water temperature. So are you suggesting warmer or colder? Mm-hmm. Are you serious? Well, that wasn't intended. Yeah, no. That's a misuse of the data, I think. Yeah, we have no intention that anybody should be heating their water up to hit our conditions. It was really just to try to get an average kind of model water temperature. So, thank you.
If you're talking to an operator of a pool and you tell them you have high cyanuric acid levels, how would you actually explain to them in a way they could understand, non-scientific way, why the cyanuric acid high levels actually slows down the chlorine, in a sense, its work? How would you explain it to them? Because it is a little bit above most people's level. If you
Well, I guess in a simple way, you'd say that the cyanuric acid combines with the chlorine and doesn't allow it to be free to disinfect. And so when it's bound up, it's not free to disinfect. So when you have the higher levels, that is sort of counterproductive. It weakens the overall.
No, it's a soldier now, it's a good one. You've got soldiers on the front lines with guns, or something like fighting the enemy. They're the active chlorine. But it doesn't matter how many soldiers you have in reserve, that's not going to have you win the war. It just lasts longer, because as your soldiers die off and stuff, you replace them, right? But in terms of the rate of killing the enemy, it's only a matter of the soldiers on the front line. And that's not the chlorine that's bound to cyanuric acid, the chlorine that's free from it. So that reserve is great for not running out of chlorine, but it doesn't help you win the war. That's the closest analogy.
Well, that can work as well. Thanks. Sorry, is there another? Yeah.
Excuse me. Did I see correctly that your tests were done at 77 degrees Fahrenheit?
25 degrees, yes.
Okay. And are the water samples that you're using, have they been adjusted for calcium, alkalinity, et cetera?
So they
Have they been adjusted to replicate a typical pool or spa?
right. So I'm not sure. We do have salts. There are salts in the water. Per the typical chemistry for producing this oxidant demand-free water. But I don't know offhand what the salt levels are that relate to conductivity or alkalinity. We don't. I will say this though, if it's a bigger issue, is that we don't typically test for those. And so if you think that kind of characterization data is important, then that's good for us to know. We're about to start a new set of experiments, and we can do that. We can try to, analyze for that if you think that's important to characterize.
Well, a suggestion from me, I don't know if anybody else agrees, but I would assume that the best way to go about this is to replicate a swimming pool environment, including temperature. We don't operate swimming pools typically at 77 degrees. So if we're trying to find out how to kill off the crypto, how to inactivate it in a swimming pool environment, that this is what we're all here for, then the studies need to be performed as such to replicate what we have to work with.
So are your pool water temperatures more?
Probably 81 to 84 degrees.
81 to
typical.
84, it's more typical. Well, I can only tell you that, yeah, so things are going to happen faster at that level. I don't know that it's going to be significantly faster, but we could maybe we could maybe up our temperatures to hit that level. So that's a fair, so again, we come here to try to provide data and educate, but obviously you all have a lot of experience and can educate us. So that's something I'll take back and we'll think about for the next set of experiments. Thank you.
Unfortunately, we've run out of time. And that is the end of the first day. I want to thank Dr. Hill for his presentation today. And I'm standing in front of the speaker. I should have known better. And see you all in the morning.